Okay, we're very happy to have with us today Dr. Sam Stevens coming up to us from the Fraternal Order of Eagles Diabetes Research Center, Department of Internal Medicine, Division of Endocrinology and Metabolism at the University of Iowa. And his talk will be beta cell to Golgi remodeling, a potential mechanism in T1D development, a little bit about the laboratory. Their lab is focused on understanding basic mechanisms of pancreatic beta cell function and survival with long-term goal of developing effective beta cell therapies for treatments of major forms of diabetes. His lab has extensive expertise in secretory protein biogenesis and trafficking, ER and Golgi function, and pancreatic beta cell function. And the PI, Sam Stevens, has been active in the diabetes field for over 17 years and is responsible for a number of novel findings. Through this work, they identified islet-derived VEGF peptide, TLQP21, as a potent insulinotropic agent able to attenuate diabetes onset, and two, develop sophisticated imaging platforms to study molecular determinants of pro-insulin sorting. This led to the discovery of ER and Golgi export defects in diabetes models. And three, identified a metabolic redox relay between the mitochondria and ER and beta cells that's necessary for insulin biosynthesis. And finally, identified cytokine-directed changes in the beta cell surface glycone that may contribute to T1D pathogenesis. They have a long-standing interest in this particular section and function of the beta cell mechanism, and really interested to hear what you have to say. Just before we jump in here, can you unpack this a little bit? The Fraternal Order of Eagles Diabetes Research Center.
Yes, yes.
What is that, center entail, and how many sort of type 1 diabetes researchers are there? If you can just tell us a little bit about it.
Yeah, so the Diabetes Research Center here at Iowa was really started with an endowment from the Fraternal Order of Eagles, right? So they did a massive nationwide campaign for fundraising, generated a tremendous amount of money for obvious interest in diabetes research, and used that as sort of the key founding block to start the center. Which I think was around 2013, 2014. So I came here in 2016. So in total, I think there's probably there's probably over 100 investigators here that are affiliated with the Diabetes Center who are, span basic science to clinical science, clinical trial work, translational stuff, across a whole series of disciplines. Type 1 diabetes actually is an area that we don't really have very many investigators. My lab and Yumi Amai's lab are really the only ones that are beta cell focused. Of course, on the clinical side, they've been involved in TrialNet and other aspects for type 1 diabetes management. But in terms of the basic science, no, that's very few investigators actually here at Iowa do this kind of work. So,
Well, it's great. I'm glad you're one of them.
yeah, thank you.
Feel free,
Yeah,
everyone. Oh, sorry, just before you jump on or continue, feel free, everyone, to drop questions in the chat as we go, and we will also answer some at the end.
absolutely. This can be as much Q&A as you want. So, yeah, so this is really actually a great time for us to present this work. I mean, you guys picked it up in bioRxiv that we put it out there. It's in review now. We actually received our reviewer comments now, which I think were pretty favorable, had some very reasonable suggestions. So I think we'll be able to hopefully get this published in the next few months. So this is great we get to sort of talk about this work a little ahead of that. So, as we alluded to, obviously my lab studies the pancreatic beta cell. These cells are responsible for glucose homeostasis through controlled release of insulin. Insulin is the only hormone in your body that really is able to promote glucose uptake, lower blood glucose. That's why in the absence of insulin, which occurs in type 1 diabetes or even insulin resistance in type 2, drive hyperglycemia. Beta cells do this by a well-established program. They sense glucose through glucose uptake and largely through mitochondrial metabolism. This generates an action potential, which provides the key signal like calcium to promote insulin exocytosis. very well established. One of the areas that my lab has traditionally been focused on has been looking at proinsulin trafficking or insulin production. And we've made some really interesting discoveries of how mitochondrial metabolism supports the redox functions of the ER. And this is really critical for proinsulin folding and proinsulin maturation to make insulin. More recently, and which is relevant to the work I'm going to talk to today, is that we've also begun to understand that in the beta cell, mitochondrial metabolism is also really critical for Golgi structure and particularly Golgi function in sort of a unique way in the beta cell that we think is highly relevant to potentially some of the early stages of autoimmune diabetes or type 1 diabetes. And so that's where I'll sort of transition to. So over the years now, initially from George Eisenbarth's model, we've kind of refined this, and we've sort of divided up the development of autoimmune or type 1 diabetes into several stages. Really, stage one, we've identified individuals who are going to have some autoantibodies, but presumably they have sufficient beta cells that you'll have very essentially a normal glycemic. But prior to that, there's quite a number of studies that have tried to look for genetic endpoints to try to identify if there's clear at-risk genes. And there's been some great work. I mean, this slide here can certainly be updated, but in general, one of the primary genes that have been associated with type 1 diabetes is in the HLA, the class 2 MHC, which of course is critical for antigen presentation. But what's rather interesting about this is it doesn't appear to be directly causative. So while the vast majority of individuals who have type 1 diabetes are carriers for one of the various SNPs in the class 2 MHC, less than 10% of the population who carries those SNPs ever develop disease. So there's a very clear disconnect between sort of the genetic risk and the development of disease. And in fact, most cases for type 1 diabetes are in fact spontaneous. They're no known family history. But one of the things that we've come to learn, particularly early in type 1 diabetes, is the presence of autoantibodies. And so a series of studies that were done, largely, I believe, in Scandinavia, have looked at individuals who have single, double, triple autoantibodies and looked at their rates of conversion to clinical type 1 diabetes. And individuals that have one autoantibody have relatively low risk, maybe 5% may develop type 1 diabetes. But if you have double or triple autoantibody, the chance is probably 80% to 100% or greater. But one of the striking findings was that even though these individuals are displaying autoantibodies, some individuals may develop T1D, within a few years of life, and some may be upwards of, 15, 20 years. So again, there's this sort of discrepancy between understanding when we have some indication that there's autoimmune activity and when we have full-blown development of disease. And so that remains one of the biggest challenges, I think, in the field is, not only identifying at-risk people, but when are they going to go through the final conversion. And so some really important studies have come out that have indicated that there's some pretty early signs for beta cell dysfunction in the development of type 1 diabetes. I think from the original Eisenbarth model, we thought it was largely just the reduction in beta cell mass. You would reach a critical phase and there wouldn't be sufficient beta cell mass to maintain glycemia. It turns out, no, that there's actually some dysfunction that's present significant, much earlier prior to disease presentation. And a lot of that's the benefits of knowing now the development of the autoantibodies. But through those types of studies, we've started to think about disease in a different way. So one, I think we often would have thought beta cells perhaps an innocent bystander of the autoimmune response. That's probably true or thought to be true in many other versions of autoimmunity. But I think now we've also come to recognize that the beta cells, perhaps their response to some of the inflammatory signals may also contribute or antagonize the immune system in a way that adds to autoimmunity. And so I think there's some really interesting areas there that we can think about. And I think that's the space this project begins to think about. So we got in on this thinking a lot about some of the early markers in autoimmunity, and that's the action of the pro-inflammatory cytokines. They've certainly been implicated for decades now as being important for type 1 diabetes. And so several actions have been known. And so this is typically IL-1 beta, TNF alpha, interferon gamma, and more recently interferon alpha. And so they have a little series of actions in the beta cell, one of which is the activation of iNOS, inducible nitric oxide synthase, which of course turns on nitric oxide. Nitric oxide has a whole host of different things it can do in the cell. In the beta cell, one of those is to suppress mitochondrial function, and this has a direct effect to block insulin release. We know that cytokines can also cause versions of ER stress, and at least long-term or in high doses can lead to apoptosis. And there's many other things that the cytokines have sort of been attributed to actions in the beta cell. But an area that kind of had we were interested in largely because of what my lab studied. A graduate student in the lab, Sandra Blom, had noticed that no one had really ever taken a look at the Golgi. We have several studies on the Golgi, which I'll sort of briefly mention. But the Golgi is kind of a critical organelle in the cell. Golgi is important for all the protein sorting of things that are headed out of the cell. So if you're going to be deposited on the plasma membrane, if you're going to be secreted, you're moving through the Golgi. So the Golgi is really a key sorting center for the cell. And so just to give you a little bit of a primer on the Golgi, because I don't think many people do think about this organelle, there's really three primary functions that I associate with it. One, as I alluded to, was cargo handling. So if proteins are destined for different cellular compartments, often it will move through the Golgi, and the Golgi is involved in that sorting. If you're going to go to the plasma membrane or the lysosome, or in the case of insulin, to a regulated vesicle, the Golgi is important for all those sorting decisions. In addition to that, the Golgi does a lot of key aspects of protein maturation, things like glycosylation, which I'll talk about, sulfation is an important modification, some of the proteolytic cleavage that enzymes like furin do. So protein maturation is also a critical function. And then an area we don't touch on, but is probably worth mentioning, is all the lipid modification. So ceramides, sphingolipids, sphingomyelins, these are absolutely critical for many different cell types, for their extracellular, for their interaction with the extracellular matrix and things like that. And so the Golgi has a key role in defining those. The other thing I should mention about the Golgi is just its general organization. So it's polar. So here on the sort of the external portion here, and it's nice to colorize here, is what we call the cis face of the Golgi. This, of course, is the accepting face. That's where ER cargo heads to the cis face. And then you move through the cisterna, through these stacks to the trans face, and then vesicles or proteins will emerge from the trans face and go on to their place. A good example would be proinsulin. Yes, Monica?
Sam, just to qualify, insulin's usually initially translated as pre-proinsulin in the cytoplasm, right?
Yep.
And then the N terminal brings it over to the translocation into the RER, and then the proinsulin folding happens in the RER. So the folding, right, and the cleavage, oxidative folding, really, and then you get your three disulfide bonds, and you get your structure. So do you think that's probably a reason why people just didn't think about the Golgi? They figured like, okay, it's all done, it's all completed by the time it enters into the Golgi and the cis face?
I think largely so, yeah. I mean, if you look at the number of papers that have studied ER stress versus Golgi stress, I mean, it's probably more than 1,000 to 1. The Golgi is a tough organelle to work on. It's tough to isolate. That it's not something I think as many labs do. It's a little amorphous as well, not that the ER isn't. And so you're right. I mean, I was taught in my cell biology classes that, the ER is kind of the rate limiting step for a lot of the protein folding. And once it happens, the rest of it just works like clockwork. And that's actually not true at all. There's quite a lot of evidence that, the Golgi is actually, if anything, it just has just a critical of a role in it. And I'll show you a little bit of evidence for that even in beta cells. So one of the things my lab had done in a separate publication is we were interested in proinsulin transport for the Golgi. And so we have been very successful making different reporters so we can watch proinsulin maturation. This is just a movie from primary mouse beta cells where we can see proinsulin that's basically fluorescently tagged as it moves from the cis face into the trans region of the Golgi. We can sort of track that over time. And here we go. And essentially what we learned in that study is we started to look at a model of insulin resistance. This is the DB-DB, the leptin receptor DB-DB model. What we found is that there were actually defects in Golgi export of proinsulin, and that contributed to some of the beta cell dysfunction that model is known to have. And when we look structurally at the Golgi are typically organized into these very long cisternal stacks. You can think of them as pancakes, really, which are kind of outlined here. And one of the things that we observed was that in the diabetes model here, that the Golgi stacks tend to be significantly shorter, and they tend to be dilated, which is often an indication that there might be sort of a problem. We also noticed that it was highly vesiculated, which again is kind of a structural defect. And this is some of the evidence that we had, at least in an insulin resistance model, where we have beta cell dysfunction, that Golgi defects could also contribute just as much as ER stress that has been looked at. But this is sort of totally separate from what we would anticipate in type 1 diabetes. So to look at this in a more type 1 relevant model, we decided to first use pro-inflammatory cytokines to see if there's any changes in Golgi structure in this model. And so Sandra Blum did a very careful read of literature to try to identify what are the relevant concentrations of cytokines. It's a wild west out there, and we zeroed in on concentrations that were consistent with what's known to be in circulation or a little bit above that might correspond to, say, a virus or some other kind of infection. And then we carefully looked at doses of the cytokines as a cocktail, IL-1 beta, TNF alpha, and interferon gamma, to look at the minimal doses that would effectively suppress insulin secretion. That's a pretty well-known phenomenon to occur. And so this is just data showing that in both a rat INS1 cell line, as well as in primary mouse islets. And so you can see this very nice sort of dose-dependent decrease in glucose-stimulated insulin secretion here, looking at a static insulin release experiment. So using those doses, then we ask the question, what about the Golgi? And so that's kind of the real meat of what I'm going to talk about. So through confocal imaging, we can look at Golgi structure pretty readily. This is in INS1 cells. The nuclei are labeled with DAPI or blue, and we can identify the cis Golgi with a marker GM130. The trans Golgi using Golgi 97. We can separate those pretty easily on the microscope. You can see the structure here. If we treat these cells overnight with an inflammatory cytokine cocktail that I talked about before, that causes a massive fragmentation of the Golgi. And these data are quantified over here. And one of the two interesting things about this, one was when we looked at the expression of the Golgi proteins, at least the ones that we've checked so far, like GM130, Golgi 97, TGN38, GRAS 55 and 65, and a few others, expression level of none of these changes, but the structure is remarkably altered. The other thing that we noted too is that when we see this fragmentation is that it looks like they're little mini satellite Golgis. So the cis and trans markers always seem to stay together. So cytokines are known can in some instances result in apoptosis, at least long term. And so John Corbett and some others have shown that cytokine effects or defects are reversible. And so we wanted to know in this model if these effects were also reversible. And so what Sandra did is she treated 18 hours with these inflammatory cytokine cocktail. We see the Golgis again go from this nice tight structure to this fragmentation. She could then remove the cytokines, put them back in growth media, and with 24 hours, the Golgi structure restores to basically to normal. So we think this is a fully reversible process. Long term, yeah, it may be more detrimental, but it's fully reversible, which is consistent with sort of actions of cytokines, and would also indicate that this is not primarily an apoptotic mechanism. If it was apoptosis, we would have thought the cells would have never survived this. So, that was INS1 cell lines. Then we moved into a primary model here, mouse beta cells from primary cells. And so mouse primary cells are a little bit different. So one is the Golgi structure. The Golgi is quite a lot bigger. It makes sense. These cells make probably 10 times more insulin than the cell culture model does, even though it's a decent model. We can still separate out cis and trans markers, GM130 and Golgi 97. We know these are beta cells because they're insulin positive. But instead of seeing this fragmentation that we saw with the INS1 cells, we actually see this sort of pronounced compaction of the Golgi. So the Golgi literally shrinks in size by almost probably 40, almost 50%, and that's quantified over here. And similar to the cell line model, this occurs without any changes in protein expression. So 18 hours of cytokines or 24 hours of cytokines, you see this massive compaction. I believe Sandra's taken this out to about 96 hours to see eventually if they fragment, and they never really do. So this seems to be the primary phenotype or the main phenotype we see in this model. Of course, this is rodents. What about humans? We see a similar compaction in humans looking at confocal, but here now we've zeroed in on the ultrastructure and done electron microscopy. And so similar to that early model that I had shown you, the Golgi stacks here, I got red arrows pointing to them because I realize not a lot of people do EM these days. But you can see these long, thin cisterna. These are very classic Golgi stacks. Lengths are easily measurable. And all the data quantified here at the bottom. And what we found is if we took human beta cells or human islets treated with cytokines and then looked specifically in beta cells at Golgi structure, we noticed they're quite disorganized. One of the main measurements that we noticed that were different is the stack length. So the length of the cisterna were quite a bit shorter. We see quite a lot of sort of disorganized vesiculation also around it, but it's a little bit more difficult to quantify. It's more of a sort of a visual inspection, but very similar to what we saw in the mouse. We see massive Golgi structural changes, and it's also consistent with what we saw in the cell line, although it's a bit more dramatic. And so to kind of fall back on, okay, so that's interesting, right? The Golgi structure change, who cares? Unless you study the Golgi. Well, again, I want to highlight the Golgi has a critical role. It is a sorting center for basically everything that's about to get out of the cell: proteins, lipids, carbohydrates, at least in terms of protein modifications, and particularly the glycosylation is a protein modification on cell surface proteins. Cell surface glycosylation is pretty critical in terms of antigen recognition, and so we zeroed in on that as something to look at to see if there was alterations. And to kind of give you a snapshot of what glycosylation really looks like, it's incredibly complex. And then in the human genome, there's over 200 glycosyl transferases that are involved in the various stages. And there's lots of different versions of glycosylation. So each one of these little building blocks is a sugar residue, and they're typically added one at a time. And so to get all these distinct linkages, that's what requires that sort of complexity of all the different enzymes. And so the basic idea that Sandra and I had was that, well, if you begin to alter the Golgi structure, maybe this sort of conveyor belt of carbohydrate assembly will also be defective. And there's evidence for that in other models that have seen some Golgi disruption. You'll see some indications that there's changes in cell surface glycosylation. And so here's just an example of this in our hands. This is in primary mouse beta cells. We use two different lectins, which are basically proteins that bind very, very specific carbohydrates. We use one known as wheat germ agglutinin, which binds sialic acid and N-acetylglucosamine. And then we also used another one called GS2, which is from soybean, which also binds a different version of N-acetylglucosamine, a different kind of setup. And so on control beta cells, we see very clear, very nice decoration of these glycosylated proteins on the cell surface. And if we treat with the cytokine cocktail, most of that WGA staining is lost. So pretty profound change in cell surface glycosylation. But it seems to be somewhat specific. If we look at, say, if we use GS2 as another way to look at different modifications, no difference at all, no difference in cytokine treated. And I think we've now looked at about a panel of 4 or 5 of these, and it's kind of mixed. We have 2 or 3 that change and a few that don't change. And so we have a lot of interest now in trying to deep dive into this in more detail to understand, again, what are the changes, and can we sort of dial that back and understand, are there specific checkpoints perhaps in the Golgi that are being altered? And so, a question though is then, so why? Why do we care about protein glycosylation? And as I kind of suggested before, protein glycosylation is absolutely a critical part of antigen presentation. And so our kind of big picture idea for this project is we think about that some of these inflammatory signals could lead to a rather profound remodeling of the beta cell surface. And that could serve in some contexts as recruitment for immune cells, either for protective reasons or potentially as a pathological mechanism. And so to give you kind of a classic example of where glycosylation is important, think about the blood groups, right? So my blood is type A, my wife is an O, I think my mom was an AB, right? We know you can't mix some of those blood groups together because you'll get rejection. The difference in the blood groups is the difference of a single sugar on the red cell surface. So I think if you're an A, I think it's an N-acetylgalactosamine. If you're a B, it's a galactose. And if it's O, you don't have either one of them. And it's just the difference of which enzyme you express. One sugar difference is enough to give you that type of difference in the blood groups. So it's not unreasonable to propose that alterations in cell surface glycosylation here could also invoke some aspect of immune response. There's a number of immune receptors that are dedicated to looking at cell surface glycosylation, fungal proteins, bacterial proteins, and things like that. Certainly would be different than self-antigens. And so there's also some indications in other autoimmune diseases like rheumatoid arthritis and lupus that glycosylation could also be important. So we think, it's certainly not the end all, the smoking gun, but could be an important aspect of modifications that could lead to further immune cell recruitment. So the other side of this then is the mechanism. How is this occurring? And so Sandra and I talked a lot about this. And so one of the classic actions of some of the cytokines like IL-1 beta is the activation of inducible nitric oxide synthase, or iNOS, which produces nitric oxide. And so we essentially wanted to know, is this action of the cytokines on the Golgi structure dependent on iNOS? And so we took wild type and iNOS knockout mice, or NOS2 knockout mice, controls. They have sort of comparable Golgi sizes. If we treat with cytokines, again, in the wild type in islets, we see this Golgi compaction. In the NOS2 islets, in the NOS2 knockouts, that's completely lost. So these data would indicate that iNOS is required for this Golgi restructuring event. We can also see this if we use an iNOS inhibitor like NMMA. One of the other things we're interested in, and this has long, I think, sort of plagued the field, is trying to understand why beta cells are the target in type 1 and alpha cells and delta cells remain. I mean, there's some indication there's maybe some dysfunction, but they're largely not the site, not the targeted cell for killing. They're usually unharmed in that sense. And so Sandra set up an experiment. This has a limitation of just a cell culture model, but I think it showcases the point, looking at a beta cell line in INS1 versus an alpha cell line in alpha TC1. So if you treat these cells, either one of these cells with cytokines, both of them clearly turn on iNOS. And in fact, the alpha TCs will turn on iNOS to a greater degree than the INS1 cells. But if you look at the response on the Golgi, they're very different. Beta cells, we see this fragmentation process that I talked about. Alpha TCs, there's no difference in size, no fragmentation that occurs. And so this was really exciting to us because this is kind of the first indication that there's some specificity in the cellular response that may also lead to differences in how cell surface remodeling is also impacted. And so to kind of understand that a little bit more mechanistically, we thought a lot about metabolism because that's an area where beta cells and alpha cells are thought to differ. So, and not to go into this too much detail for people who don't understand metabolism, but you can kind of think about this, the upward phase of glycolysis, and then the second aspect is the TCA cycle. So beta cells primarily will take glucose through glycolysis and all the way through the TCA cycle, and that's how they make ATP. If you shut down the mitochondria, they can't use this top phase anaerobic metabolism. They'll just shut down. Most cell types actually can do the switch. They can shut down mitochondrial metabolism and then primarily generate ATP anaerobically through glycolysis and through production of lactate. Beta cells have very, very little to no expression of the enzyme lactate dehydrogenase, so they can't really do this effectively. And that's important because this is actually important to their mechanism of glucose sensing for insulin secretion. So John Corbett had some really interesting data sort of showcasing this between different cell types. And so nitric oxide is very well known to block cis conatase in the TCA cycle. And so he used a chemical donor of NO known as DPTA and could essentially show that an INS1 or a beta cell is very sensitive to NO, where you can shut down ATP production, you can shut down NAD+, versus a mouse embryonic fibroblast, for example, is just chugging right along because they can switch to use the upper ends of glycolysis for ATP. So that cell type specificity becomes important. And so then our question became, well, is it the beta cell sort of reliance on mitochondrial metabolism? Is that really a unique sensitivity that they have to cytokines and NO signaling? And so to look at the role, whether or not metabolism directly contributes, we could use here a very minimal media. It's kind of what we think of as like a Krebs ringer media. There's no fuel in there aside from the glucose that we've added. Under those conditions, we see pretty classic Golgi structure. If you omit all fuels, including glucose, you see really rapid fragmentation of the Golgi, which would again suggest that the metabolic fuel is important. By the same token, you can take cells and culture them, treat them for a short period of time with rotenone, which is mitochondrial inhibitor. That's sufficient to cause really substantial Golgi fragmentation. So again, beta cells really you need active metabolism to maintain Golgi structure. That's actually true for most cell types, but the beta cell specific in the sense that it needs mitochondrial function. And so sort of the last bit of data that I'm going to show you, Sandra thought she would test this in a few different models or a few different cell lines. So we have the alpha TCs as an alpha cell, generic HEC293 cell line, and then the INS1 as our beta cell line. So here we use DPTA as a nitric oxide donor. And so in this model, beta cells are the only one that are sensitive to DPTA. This drives Golgi fragmentation. 293s and alpha TCs are totally refractory to this. We can also culture these cells in the absence of glucose and see no changes in Golgi structure. A little different than the experiment I just showed you because this is in a complete growth media. So these cells have access to amino acids and other things that can enter the mitochondria. And beta cells here, and that's probably why the beta cells here don't fragment the Golgi. But if you remove glucose, so they have basically no fuel here, and you'll simultaneously block mitochondria using DPTA, in all the cell types now, we unveil this sensitivity. And so we think that the reason the beta cell is very specifically targeted in terms of the cytokines is because of the reliance on mitochondria, while the other cell types can maintain this glycolytic capacity to maintain ATP. And so just kind of quickly summarize what I showed you. Proinflammatory cytokines have this reversible ability to impact Golgi structure. It certainly appears to be iNOS and NO-dependent. And this, we think, is mediated through the mitochondrial metabolism, which is really fascinating because of the beta cell specificity there. And these alterations, we think, can lead to changes in the cell surface. Why are these things important? What are our next steps? Well, one is to better understand how the inflammatory cytokines actually act on the Golgi. We have a couple of target proteins in hand that we're following up, which we think might regulate that Golgi structural change. Of course, we want to really understand in more detail what's the impact of the cytokines on the beta cell surface remodeling. So we're doing some work with John Corbett's group to try to look at some beta cell surface markers to see if those are changed. Probably one of the most important questions is whether or not these coincide with development of inflammation in type 1. I mean, of course, I recognize these are sort of all cell culture-based models. So we're doing studies now with NPOD to do staining to see if we can correlate structural changes or not with the onset of type 1 diabetes. We're also using a site called Nanotomy to look at EMs to also do those correlations. And then I think the last question, which I think is the most provocative to me, is are the changes in the surface remodeling, is that a pathological event that sort of contributes to disease development, or is it used as a protective mechanism? We know that viruses use glycoproteins for cell entry. So maybe this is a means that immune system senses viral infection of some of the cells. The cytokines are then used to sort of reprogram, remodel the cell surface to prevent the other cells from being infected. Certainly there's longstanding ideas about how viral infection is important or might contribute in type 1 diabetes. So we always kind of keep it in the back of our mind that this may be protective and adaptive, or it could be maladaptive. With that, I'll sort of thank people in the lab that contributed. Largely, probably 99% of this work is Sandra Blom, graduate student. If you're looking for a postdoc, I'd be happy to put you in touch with her. She's going to graduate this June and has been a phenomenal student, as you can see by the project. Through Sandra's work, we actually are funded by Breakthrough Type 1 Diabetes on this work. Palin Narcian's an undergraduate in the lab who's been helping out quite a lot. This is a collaboration with Ling Yang, who has the NOS2 mice, and then John Corbett, who's sort of an expert in iNOS and is going to help us do some cell surface stuff. And then James Ankram's lab with Riley also helped us do some work that was important for this project. And then the last thing, of course, for all the human islet work we do, we can't do it without donors, without that donor tissue. That's absolutely essential for us to be able to test this in human models. So with that, I can stop and answer any questions.
Thank you, Sam. That was really, really interesting. We do have a question in the chat. Shamik, do you want to unmute yourself and ask it?
Hi. From your EM studies, it seems like there is a mitochondrial division defect or something like that on addition of cytokines. Could you please, is it correct or I'm overjudging it?
No, I think you're correct. I would be surprised if there was not mitochondrial structural changes. I'm pretty sure that I think Scott Solomon Poor has a paper where they have looked at that as well, and there's definitely some structural changes that occur with it. I don't know mechanistically. I don't know that it's understood how or why that's occurring, but yeah, we didn't carefully look at it in our EMs, but I'm pretty certain that it's probably there. Yeah.
Thank you. Anyone else? Questions? Drop it in the chat or raise your hand. Feel free to unmute yourself. I had a quick question. I noticed you guys were using the TNF alpha, but if you do a quick search, basically how different viruses affect the beta cell Golgi, you come up with the fact that enteroviruses, have a structural disintegration of Golgi, such as, CVB, alpha viruses, more like influenza or encephalitis, something, those do membrane repurposing, and then the coronaviruses do functional impairment. So, have you thought about actually maybe collaborating with someone who does viral, up one step upstream from the TNF alpha, right? Who does viral studies and seeing if these different virals play a different role, or even if different concentrations of the TNF alpha make a difference.
Yeah, no, that's an excellent question. We have thought about it. I think there's a lot of areas that we could go into, and I think that one would be pretty important. We haven't done it yet. I'm actually on a thesis committee with one of John Corbett's students, and John's lab actually has been looking at how cytokines can sort of impact viral uptake, viral entry, viral replication. And you're right, several of those viruses actually will hijack the Golgi for their own purposes, largely for viral replication. And so we've thought about it. We haven't done any experiments to test it, but I think it would be really interesting to do. I think also in the thoughts too about particularly about the glycoproteins and whether or not that may be used as a way that the cell protects itself by removing some of the viral entry receptors too. So yeah, very interesting.
Yeah, it's so many interesting places to go from here. Here's a couple of questions from Jemming Liu at City of Hope, saying, great talk. Does interferon alpha also induce Golgi alterations?
To be determined. We're
Okay.
doing it, but don't know, but that's a great question. So yeah, we're looking at that. Yep.
And also, does cytokine-induced Golgi alteration promote HLA class 1 expression in beta cells or a compensatory effect to autoinflammation?
Another great question. That's actually something that we're looking at as well. I mean, I know there's some really nice data now on interferon alpha's ability to increase class 1 expression. And so, yeah, we're doing those studies as well. We're trying to also tease out, are there different aspects of the signaling between the cytokines that contribute in either different ways or additive ways. And so I think that'll be really important also to figure out. Yeah.
Hmm. Yeah, it's really interesting. And it looks like Sandra Blum will be entering the postdoc market soon. So just a shout out to anyone who's interested to reach out to you, Sam. Last call for questions. All right, we look forward to posting this on our website and our YouTube. And thank you again for sharing this really interesting work. It's great.
Thanks.
Oh,
Thanks so
hold
much.
on, one more. Kiadi?
Yeah, there is one question I had. Like, means when you give the cytokine treatment, there's diffusion of the Golgi, right? And in different mouse model and islets, they contract. So can you actually determine using assay like bioID or something to understand what proteins are actually diffusing, and this diffused protein, what are they actually interacting with, which may be leading to this kind of phenotype? Like,
Yeah, that's a yeah. So I mean, there's a lot to unpack there. So first, yeah, why the difference between like the cell culture model, the INS1 cell and the primary cell? It's not something we understand. Like phenotypically, the other outcomes, the other endpoints we've looked at seem to be consistent between them, but the structural change is sort of different. Maybe a dividing cell versus a non-dividing cell. I'm not totally sure. In terms of identifying the proteins that are responsible. So we've had a couple of successes just in our query of proteins that are normally Golgi localized, and found some proteins that change, and we've been doing knockdowns to see which ones might be responsible. We've also through Ling Yang and some online resources have looked to see, are there certain Golgi proteins that are modified by nitrosation. And we have some candidates there as well, where we're again following up to see if they might be important. Um, your question's also interesting, that we could expand that and do sort of a larger screen. Basically, I mean, we can pull down Golgi as a full structure and see, okay, yeah, are there things that are modified there, enzymes that might be regulatory that could be important. Yeah, it's certainly an area that we can explore. We just haven't done it yet. But yeah.
thank you.
And then just from PLE, really great work. Looking forward to seeing where this goes and what we can start looking for in the membrane. Although I think FFPE tissue may give you some headaches for the smiley.
It already has. My undergraduate Pauline has taken on this sort of matrix of trying to get good staining. So we're also going to do some cryosections and see. I mean, that's been one of the challenges. The Nanotomy website, which are EMs, that Sandra's painstakingly going through now to also look at Golgi structural changes, may turn out to be the slightly easier route to go, because you're right. I mean, doing you identify cells and nuclei and stuff like that with paraffin sections, but doing subcellular structure like that is a lot more challenging. So we're also thinking about this, and NOD is another model that we could sort of do this in. So yeah. With this luck, and any suggestions and people have on how to do this would be helpful, because yeah, this is it's a challenge.
Yeah. Oh, looks like there's a reply. If the elite lab can help from England with the FFPE, they're happy to chat. They're at Exeter.
That would be fantastic. That would be fantastic. Yeah. I mean, we've gone through sort of our standard changes of antigen retrieval and things like that. But yeah, it would be good to discuss, and maybe, yeah, I may follow you up on that for in the near future.
One more from La Jolla, Medibuncala. We've observed an accumulation of HLA-1 in GM130 in autoantibody positive and T1D, but not in controls. Wondering which dysfunction comes first, either ER or Golgi.
Yeah, chicken and the egg for sure. Yeah, I mean, the ER stress is really interesting, particularly in the cytokine space, because there's some really big differences in, like, how we view canonical ER stress if you use like fapsigargan, like a circuit inhibition versus palmitate versus cytokines, where there's some overlap, but there's really very clear differences. I mean, I think Ross Labut has some nice data that showcases, where they begin to diverge and some things like that. As far as I know, I mean, Carmela Evans Molina group published a nice bioinformatics paper in diabetes showing, that there's some signatures of Golgi stress. But yeah, I mean, we're doing studies now in our hands. What we see for ER stress looks quite different than what we see here in the cytokine model. But we're doing some studies now to really see if we could separate the two out. But in terms of which comes first? I mean, Profeldin will cause ER stress. And I'm sure some ER stress will activate Golgi response. So yeah, that is a tough question. Yeah.
Thank you, Sam. This really sparked a lot of discussion, and I'm sure there's going to be some follow-up communications and hopefully some collaborations to really dig in here. It seems like there's a lot of exciting work to be done. So thank you so much again, everyone, for contributing their ideas, and thanks again, Sam. We'll keep in touch, and we'll be looking for the paper.
All right. Thank you so much, Monica. And also shout out to Alexis, whom I met at InPod a few weeks ago. Yeah.
That's great. Yes, she's a critical force at Sugar Science, and she's here on the call.
Fantastic. All right. Take care.
Thank you. Bye-bye.